Let's jump right in. Today's explainer is all about the anatomy of an invader. We're exploring the fascinating, almost mechanical world of the ultimate biological hijacker, the virus. Now, we've all battled them at some point, right? But when you actually look closely at how they operate, it really feels less like biology and way more like a microscopic sci-fi thriller.
Which brings up a truly wild question. How exactly does something without a single cell take over the world? It's literally incredible to think about. Today, we're going to explore how an entity completely missing the fundamental building blocks of life still manages to dominate incredibly complex living organisms, completely taking over their internal machinery with ruthless efficiency.
Here's our dossier for today. We'll meet the ultimate parasite, look at viral shapes and sizes, peek inside the viral structure, check out bacterial hijacking through phage cycles, and finally, dive into animal cell invasions. Let's get to it.
Meet the ultimate parasite, the missing pieces of life. To really grasp how they invade, we first have to understand what they're fundamentally missing.
By definition, a virus is an obligatory intracellular parasite. The absolutely crucial word here is obligatory. Unlike bacteria, which can kind of just multiply on their own if the environment is right, a virus absolutely requires a host cell to multiply. Without a host, a virus is completely inert. It can't grow, it can't move, it can't do anything. It's really just a dormant package waiting for a door to unlock.
So why are they so dependent? Well, because unlike the cells that make up you, me, or even a plant, viruses are incredibly minimalist. I mean, they completely lack a plasma membrane. There's no cell wall, and the cytoplasm, that jelly-like substance where the actual business of cellular life happens, is completely absent. They bring absolutely no life-sustaining machinery of their own. Zero.
And yet, despite missing literally all of these living components, they can infect virtually any organism on the planet. We're talking everything from invertebrates and animals to plants, fungi, and even bacteria. The only catch? Their host range is strictly determined by attachment sites. If the structural Ts on the virus happen to match the receptor locks on the host cell, well, they're getting in.
Moving on to viral shapes and sizes, the scale of the threat. Let's look at their physical scale to understand just how stealthy these invaders actually are.
To put this into perspective, their size varies from a minuscule 10 to 1,000 nanometers in length. For context, a single nanometer is 1/1000th of a meter. That makes viruses completely invisible to standard light microscopes. They're operating on a scale so profoundly tiny that they can easily slip past our macroscopic defenses completely undetected.
And within that incredibly tiny scale, there is a sheer diversity of forms. Categorized into DNA and RNA families, they aren't just one standard shape. From simple, tiny spheres like the parvovirus to complex thread-like structures like the filovirus, their architecture is super varied, yet perfectly engineered for their specific host targets.
Now, let's look inside the viral structure at their armor and payload. We need to zoom in even closer and unpack the invader's gear to see what they actually carry.
Remember how we just said they lack cellular machinery? Well, what they do bring is a highly efficient payload. This consists of a single type of nucleic acid. It's either DNA or RNA, but never both. This genetic code can be double-stranded or single-stranded, linear or circular. And to keep this precious little blueprint safe, it's housed securely inside a protective protein code called a capsid, which is itself built from smaller subunits called capsomers.
So how does this all build into their final form? Some are what we call naked viruses, which is essentially just the genome and the capsid. Others are enveloped viruses, meaning they've actually stolen a layer of lipids, carbohydrates, and proteins from their previous host cell to basically cloak themselves. And it's amazing how these capsids assemble into striking geometric shapes. You've got the 20-sided icosahedral, the tube-like helical, and those wild lunar lander-looking complex structures.
Next up, bacterial hijacking, phage cycles. How exactly do they use this geometric gear? Let's look at a specific type of virus called a bacteriophage, a virus that strictly infects bacteria, to see how they hijack these cellular factories. This is a classic smash and grab routine known as the lytic cycle. It moves aggressively through 5 distinct stages, attaching to the host, penetrating its armor, forcing the biosynthesis of viral parts, maturing those parts into brand new viruses, and finally, releasing them by totally destroying the host cell.
You can really see the mechanics in action during an infection. The complex phage lands on the bacterial cell wall and literally acts like a microscopic syringe during the penetration phase, injecting its DNA straight into the cell.
This immediately triggers biosynthesis. The bacterial factory is completely taken over, its own operations are shut down, and it's suddenly forced to manufacture viral heads, tails, and DNA until the cell just bursts. But not all viral invasions are loud and explosive right away. We really have to contrast the explosive lytic cycle, where the cell immediately lyses, with a much more stealthy approach. This one is known as the lysogenic cycle.
In this lysogenic cycle, the viral DNA acts as a sleeper agent. Instead of destroying the cell right off the bat, the viral DNA integrates quietly into the bacterial chromosome, becoming what's called a prophage. The bacterium then just reproduces normally, copying the viral code along with its own, generation after generation, completely unaware. That is, until some external event triggers a sudden lytic breakout.
Finally, animal cell invasions, the takeover. Bacteria are single-celled organisms, so let's see how this whole strategy adapts inside the much more complex cells of animals and humans.
Taking a look at a DNA virus, like papovirus for example, the mechanics change slightly from the bacteriophage. Here, the entire virion actually enters the host cell. It travels right to the nucleus where it uncoats its DNA. From there, it ruthlessly hijacks the host's own nuclear machinery. It transcribes viral proteins, replicates its DNA, synthesizes capsid parts, and finally assembles and releases mature virions.
RNA viruses, however, play by slightly different rules. Because they use RNA instead of DNA, they often skip the nucleus entirely and multiply right within the host's cytoplasm. To pull this off, they bring or create a highly specialized tool, a viral RNA-dependent RNA polymerase. This allows them to rapidly synthesize their own proteins right on the main floor of the cellular factory.
It's fascinating how RNA viruses adapt their code to do this. Positive sense strands are incredibly efficient. They act just like cellular mRNA and get translated directly into proteins almost immediately upon entry. But negative sense strands and double-stranded RNA, well, they have a hurdle. They have to manufacture that positive strand first using that specialized polymerase before they can begin taking over the factory. It's just an amazing molecular workaround.
When you step back and look at the whole picture we've covered in this explainer, it truly is a masterclass in biological efficiency. Think about it, viruses bring absolutely nothing but a genetic blueprint and an armored shell. Yet they possess the exact structural keys to enter, entirely reprogram, and ultimately dominate a living, breathing cellular factory.
I'll leave you with this final thought to ponder. Considering that viruses completely lack cytoplasm, cell walls, and the inherent ability to multiply on their own without a host, are they truly living invaders, or are they just the universe's most highly efficient, autonomous, and rogue pieces of genetic code? Something to think about the next time you're fighting off a bug. Thanks for joining me for this explainer, and I hope this helps you visualize the fascinating microscopic world operating right under our noses.